Hi, so good afternoon and good evening, everyone. My name is Adrian Gilbert, and I'm one of the new co-chairs of the Scientific Advisory Board. As Faye mentioned, after many years of valuable service, Mike actually has decided to step down as chairman, Mike Brownstein. And I want to thank Mike personally and on behalf of the whole Scientific Advisory Board for his outstanding and sustained contribution. I know that he will remain very active in pursuit of the therapies for FD. So just to explain, earlier this year, the Foundation appointed two new co-chairs with very different backgrounds to take the forward. And here we are. Firstly, I'd like to introduce Professor Francis Lefcourt, who many of you know is a professor of cell biology and neuroscience at Montana State University in Bozeman and has published extensively about FD. Frances has a remarkably successful record of groundbreaking research in neurological disorders, and we are very lucky to have her. And in contrast, I'm actually a chemist by training. I'm located here in sunny Israel, although the sun is just going down, and with more than 2 decades in new drug development at Teva. I actually now head the pharmaceutical development at Neuroderm, which is an Israeli-Japanese company, part of Mitsubishi Tanabe, and we're developing new therapies for Parkinson's disease. And my eldest daughter, Tamar, who's watching, so hi, Tamar, is 19 years old and has FD, and I was also head of the Israeli FD organization some years ago. So we bring in a very different but very complementary background to our new roles with a real mix of academia and industry. But we do share a strong and drive and commitment to improve the lives of all of our patients and all of those with FD who are watching, and to find those new treatments and just as soon as we can. So going forward, as you can see from this slide, several of our key contributors are on the SAB we're going to continue, and in particular Horacio and Sue and Lucy and others. But I would also like to thank publicly all those who've contributed to date and are handing the baton over to some new faces. And as you can see, we've brought in some new people, and I would like to introduce them to you briefly. Firstly, there's Adam Sachs, who, great guy and a parent like me, and I'm sure many of the US families know him well. He's a leader at Paragon, which is part of Catalent Biologicals, which is actually one of the top gene therapy companies in the world. So he's a great asset. We've also brought on board two renowned discovery scientists. One's an academic, who Horacio mentioned, Professor Adrian Craner from Cold Spring Harbor Laboratory, and from industry, a friend, Dr. Ralph Laufer, who's ex-Merck and Teva, who's actually now at a company called Lysogene, working on gene therapy for rare diseases based in Paris. So these are two great additions who are going to really bring us a new dimension to some of our thoughts on how we can move forward. And our final recruit is Dr. Bat El Baraluma, who you've just met, from the FD Treatment Center in Tel HaShomer, and she also brings, obviously, her expertise in pediatric pulmonology. We have a big challenge, and I've tried to capture here 4 of the key areas I think where we're going to be focusing as we move forward. And one of the big challenges today, obviously, given the funding concerns, the way that we can actually connect with our patients in our centers, is basically how we can build new partnerships and new directions and get those new therapies out there to make a difference. So this is us, and this is what we're going to be doing moving forward. I'm incredibly excited to be involved and to be this, and indeed honored. And I'd like to now hand over to my colleague, Franz, to introduce today's scientific research presentations.
Hi, everyone, and happy FD Day. Thank you so much, Adrian, for that introduction. Like you, I share a lot of excitement about our new scientific advisory board. I'm also just so happy to be co-directing it with you. I want to add that the major reason why my lab works on FD is because I'm also part of the FD family. I have 2 first cousins in Canada who have FD. So I'm as eager as all of you for developing new treatment strategies. The good news is more and more people are engaged in clinical and basic FD research, and I think the horizon looks bright for new treatment strategies for FD. So I'd like to introduce the research session now. And first, there's an announcement just to make sure we're on the same page. So if I could have the next slide, please. Okay, so there's been a name change. The gene that Sue Slagenhop cloned for all of us originally was called IKB-KAP, which is a mouthful. So there's been sort of a convention change, and now the gene is called ELP1. And the protein encoded by that gene, we used to refer to it as ICAP, and now we're calling it ELP1, but you'll still see that those names are used interchangeably. So as you all know, in FD, the problem is that this protein, ELP1, its levels are reduced, especially in neurons, and there's some nice looking neurons there to the right. And so the goal for all of us is to try to increase ELP1 in neurons. Okay, could I have the next slide, please? Okay, so we have 4 brief talks today that will summarize research projects on FD. And of course, there are many other labs working on FD, so this is just a snapshot. But all 4 of these studies are preclinical. And all of us, as you'll see, are really aiming to translate our findings into the clinic. So our first speaker will be someone you all know, Dr. Lucy Norcliffe-Kaufman, who's the co-director of the Dysautonomia Center at NYU. And she'll be telling us about her work on the natural history study. And she's also generated a clinical development plan. And I hope you realize that these studies are absolutely fundamental for all of the clinical research the rest of us are doing. We need that foundation for our studies. Next, we will have Dr. Susan Slagenhop with us, who's a professor of neurology at Massachusetts General Hospital and at Harvard Medical School. She's also the scientific director of the Massachusetts General Research Institute, and she'll be describing her work on using small molecules to increase ELP1 levels in FD. I'm a professor of neuroscience at Montana State University and will be speaking next after Sue. And I'll be summarizing 2 projects going on in our lab, one on trying to prevent the progressive loss of vision, so the optic neuropathy that often develops as patients age. And the second project is we're trying to understand the role of the gut bacteria, the gut microbiome, and metabolism in FD. And the last talk in our session will be from our valued Israeli colleagues, Dr. Miguel Weil, professor at Tel Aviv University, and Dr. Bat El Bar Olumav, who's at Sheba Medical Center at Tel HaShomer, and they'll be presenting their work on using vitamin D derivatives to improve the health of neurons in FD. So I wanted to also bring your attention to the fact that there is a booklet that the Dysautonomia Center put out that will summarize much of this research, and it's on the Foundation website, so you can access that there. So it's my pleasure to introduce our first speaker in the session, Dr. Lucy Norcliffe-Kaufman from NYU.
Hi, good morning, good afternoon, or good evening, depending on where you're tuning in from. My name is Lucy Norcliffe-Kaufman, and I'm going to tell you about the natural history study we have for patients with familial dysautonomia. A natural history study is a study that follows a group of people over time that have a specific condition or disease, in this case FD, and it collects health information to answer pressing questions like how a disease develops and how best to treat it. A natural history study forms part of a clinical development plan, which is really a blueprint for how new therapies can be tested in the clinic to target things that really matter to patients. FD was first described around the 1950s. For the next 20 years, there was really a flurry of information describing the clinical features of the disease. The first FD database was created by Dr. Axelrod around 1969. And around 2014, we modernized the database so it meets our needs today, and we can use it to develop guidelines that shape clinical practice, but as well in looking at towards the future with our clinical development plan, which meets patients with FD where they're at today and helps us think in ways in which we can find and test effective new therapies. So you're probably wondering, how does a bunch of data locked up in a cloud help me as a person with FD? When you come into the clinic, you meet with our nurse practitioners who collect a lot of vital health information, but you also meet with our FD scientists that use surveys and questionnaires to track features over time. All of this information comes in through our clinical research team and is uploaded into our natural history database, where it's used not only to shape guidelines, but to help make clinical decisions on a day-to-day basis. But in addition to this, when you come into the clinic, you also have the opportunity to give a sample of blood alongside your clinical information. And this blood is shared with our team of basic scientists who are all working on FD. That includes Dr. Francis Lefcourt, who's using mice models to tackle the neurological underpinnings of FD, Dr. Sue Slagenhardt, who's looking at the genetic underpinnings of FD, and Dr. Miguel Wild, who's taking cells in culture and looking at the effects of different vitamins. Now, all of that information comes together with the idea of potential new therapies and options that we can try for patients. But basic science doesn't happen in a vacuum, and all of the basic science information is collected and considered alongside the clinical data. So we can link it back to individual things that happen to particular patients, but also to make sure we're targeting relevant pathways. And together with our scientific advisory board, we've really created a mechanism where drugs ideas can be tested with the hope that we can bring them back into the clinic and help the lives of FD patients today. One of the things that we're very excited about with the natural history study is that we're now working with a team of global investigators who all have permission for their patient data to be included in the database. So the steps are really in place to make this a global effort for every patient to get involved. We currently have 93 patients enrolled in the natural history study, which means there are 222 patients with FD out there that are not yet part of the natural history. What we want to do is bring those patients into the study because our data set becomes much more powerful if we're all together. This is a map of the Pale of Settlement that shows the Jewish emigration patterns. And what you can see is the vast majority of people emigrated and went to the US. The vast majority of people in our database are from the US, but we are still missing 45% of the US FD population. In Central and South America, we're still missing 2/3 of people. In Canada, however, and this is a shout out to the Canadian FD Foundation, we are missing only 16% of patients. In the UK, we're missing 82% of patients. And in Israel, we're still missing data from 98% of FD patients. So I really hope that this is an opportunity for us to galvanize the advocacy community to get behind these projects, to make sure that we can include people in the study regardless of where they live. If we look at the age breakdown of the patients enrolled in the natural history study, we see a few interesting things. We have 38 pediatric patients that we follow within our clinic, but the vast majority of patients are actually in the adult age range, and we know that there are different needs at various stages of the disease. Now, how can we give as many people as possible the chance to try a potential new therapy? Well, that comes from a thorough understanding of the underpinnings of FD. We know from the work of Dr. Francis Lefcourt and Dr. Sue Slagenhardt that there is this stage in development where ICAP is critically important, but there's also a stage, particularly through the teenage years, in which the nerve cells start dying off. And then, on top of this, in the adult patients, we start to see the effect of wear and tear on the organs, with things like high blood pressure and problems with the eyes contributing to some of the features. So it may well be that we need different things to measure at different stages of the disease. In younger patients, we might be looking at the changes in the retina, whereas in older patients, we might be looking at what happens with their walking. So what can you do to help as a community? The first thing we really would like you to do is to register and get involved in our natural history study, which you can do either by visiting one of our registered sites or calling us on the phone where you can consent to be involved. Another thing we need you to start doing is to start sharing your voice. Very soon, we're going to be going to the Food and Drug Administration, where you as FD patients and family members have the opportunity to tell your story about what matters to you. So hopefully, I've convinced you that the natural history study is a very important study that supports clinical care, basic science, and the path forward towards new therapies. You can participate in person at Hadassah, NYU, or Tel HaShomer, or call us and participate virtually over the phone. But we want you to share your voice and to come with us to tell the FDA what bothers you and why you need new therapies earlier. You can stay in touch. We regularly post updates from our natural history study on our website, the www.dysautonomiacenter.com. And finally, I want to thank Dr. Maria Kotrina, who's been instrumental in taking this project where it is today and really giving us the possibility to have an international, highly collaborative effort. Thank you.
Thank you so much, Lucy. That's such an important message. And, you know, we really are a community and we need everyone to participate so we can all move forward together. Well, now it's my great pleasure to introduce Dr. Susan Sloganhoff from Mass General Hospital and Harvard Medical School.
Hi. I'm really, really pleased to be here today for our first virtual FD Day. And while I miss seeing all of you in person, I'm really excited that we are able to include our friends from around the world and all of our friends in Israel. So I think that it's a unique opportunity for all of us to get together. So as many of you know, my lab has been working on FD for nearly 30 years now. And this is just a picture of the lab team that works on FD. And I'm going to tell you a little bit about the work that we've been doing in the Mass General Research Institute in the Center for Genomic Medicine. And so before we get started, I have to give you a little genetics lesson. And so I want to start by just explaining to you about your genes. So your genes are located along a strand of DNA. So here's the string of DNA. And these genes are broken into pieces called exons. And in order for the protein to be made correctly, these exons have to be assembled correctly. So the instructions for proteins contain these exons and they have to be put together correctly. So in this example, it has to go blue, green, red, light blue, yellow in order for the protein to be made correctly. So it's kind of like thinking about a train and train cars. So if you look down below, you have a series of train cars. And let's just pretend that in order for this train to actually be able to get to the target, the train cars have to be assembled, toys, then juice, then milk. You can't leave out the juice car, or you can't flip the juice car with the toys car. It has to be assembled in just this way in order for the train to actually be able to run on the tracks. And that's the same way with your genes. So we know that the DNA change that causes FD disrupts a gene called ELP1. And what we see here is that it is a T to C change, so it's a C here that should be a T that causes FD. And what happens when this C is present is that exon 20, so remember the train cars, this is supposed to be put together, exon 19 followed by 20 followed by 21. If this comes together properly, then we get ELP1 protein made. If exon 20 gets left out, when the C is present, sometimes it skips exon 20. If it gets left out, then we don't have any ELP1 protein made. So what happens in FD? In FD, again, we want the trains to be put together 19, 20, 21. But in FD, most of the time, exon 20 is left out. And so that's what I'm showing here. Most of the time, exon 20 is left out. And that results in ELP1 protein being very low. And in fact, we see low ELP1 protein in almost all tissues, but in particularly neurons. And so cells in the brain and the peripheral nervous system do not have enough ELP1 protein. So how can we treat FD? We need to find a way to increase exon 20 inclusion, right? So it seems like a pretty simple problem. What we want to do is make these instructions for the gene to make ELP1 protein go together correctly. So we want to make exon 20 be put in the right place. And so there are many ways we could do this. And my lab has been working on developing a drug that when we put it on cells and give it to animals and eventually to people, it will cause exon 20 to be included. And if we can increase the level of exon 20, then we will end up increasing the amount of ELP1 protein, which is what we need to do in FD. There are other ways to do this as well, and that is using other pieces of DNA that could be turned into drugs. And you may have heard of ASO, antisense oligonucleotides, or modified something called a U1 snRNA. These are pieces of DNAs that are made into drugs that could also increase the level of exon 20. So our goal, of course, is to increase the amount of ELP1 protein in patients. So my lab has been working on developing a drug for FD. So back in 2005, we found that a kinetin, which is a small molecule, a drug, can increase the amount of exon 20 in the gene. And we found this by studying FD cells in a dish. And then we spent many years trying to create a mouse that has FD. So we do a lot of work with this mouse in my lab. We worked with Dr. Dragatsis and Dr. Dietrich at the University of Tennessee, and we created this mouse in 2015. And this is a very special mouse because not only does it have FD, it also has the human gene. So it has those train cars, exon 19, 20, 21, and it leaves out exon 20, just like FD patients do. So this is a very important mouse because we can treat the mouse and see if we can increase the inclusion of exon 20. So a couple years ago, we were able to show that if we give kinetin to our FD mice, it increases ELP1 protein and it increases the number of neurons, so brain cells and peripheral nerve cells, and it improves motor coordination and spinal curvature. So this is an exciting result. Now, many of you know that we've been working on this for quite a long time, and we learned that kinetin is not a great drug. It's too weak. It's not potent enough. It doesn't do a good enough job for people. So in 2012, we started a project funded by the NINDS Blueprint for Neuroscience Research Program, and to identify a better kinetin, something we called superkinetin, a drug that would be much better at increasing exon 20. And then in 2015, we formed a partnership with PTC Therapeutics, a company that's focused on finding drugs to modify this RNA splicing. So together with PTC, we have developed an excellent drug candidate, an excellent superkinetin that is currently in what we call late-stage toxicity testing. So although PTC is no longer bringing this drug to market or bringing this drug to clinical trials, they have agreed to continue to work with us and to provide this drug to us so that we could continue developing the drug. And so late-stage toxicity testing means we're testing to make sure that it's safe enough for patients. So if the drug proves to be safe, then my team, along with help from PTC and in collaboration with the FD Foundation and the FD Center, will all work together to hopefully bring the drug to clinical trials within 1 to 2 years. So what else are we doing at Mass General? And again, this is the team that's hard at work. They're all here today watching FD Day, and I'm really glad that they're here to see you virtually. But we have a lot of other work going on in the lab. And the point I wanted to make today is that it's all collaborative, meaning we're working with all of the other scientists that you're going to hear from today to try to make these projects, to bring these projects forward. So we're characterizing the eyes of those FD mice I showed you to understand the blindness. We're working with Dr. Lefcourt's lab on gene therapy, on developing gene therapy to hopefully treat blindness. And we're also working with a lab in Italy of Dr. Pagani to understand other new ways to maybe treat the eye using different ways to increase the level of exon 20. We're still working on developing the drug I told you about, the superkinetin, and we're currently testing that in our FD mice. And I just want to leave you with that message that we're always working with all of our colleagues for ways to find treatments for FD. And we need you. As you heard from Dr. Lucy Kaufman, we need to all work together. And most importantly, you are our partners in this work. We need you to be ready to participate in trials and to participate in the natural history study. Because when any of these drugs is ready to go, we need you to be ready to go as well. So thank you, and I look forward to answering any questions.
Thank you so much, Sue, for that excellent talk and for the brilliant work that you and your fantastic team do. We're really grateful. So now I'm going to introduce my own lab's presentation. Where our lab is at Montana State University. My team is also tuned in, and we've been studying FD for over 10 years. And so if I can have the presentation, please. Hi, everyone. This is Frances Lefcourt, and I'm going to be talking about some of the therapeutic strategies that we're using in our lab now to try to improve the health of people with FD. One of the projects I'm going to be talking about involves the GI tract, gastrointestinal function. So I thought it'd be nice to show you a photo, an image taken by Marta Javert in my lab of what neurons in your gut actually look like. And that's shown here. Okay, so the first of the two projects I'm going to talk about is a project we're doing that is called gene therapy. And basically what gene therapy is, it's the use of a virus to introduce a healthy copy of the gene that you want to replace. So in our case, we know we have a faulty copy of the ELP1 gene. And so we want a virus that will introduce a healthy copy of ELP1. Now, we want to target the retina because we know loss of vision is a major problem for people with FD as they age. And a picture of the eye is shown here. And in the back of the eye is the retina. And that's illustrated here. So the retina is composed of a set of different types of neurons. And from very nice work done in the Dysautonomia Center, we know that it's these large retinal ganglion cells that are the neurons that die in FD. And those are the neurons we want to rescue using gene therapy. So what we're going to do is we are using our mouse models for FD in which we have the same kind of progressive loss of retinal ganglion cells in the mice that occur in people with FD. And so we've made a virus that encodes a healthy copy of ELP1, and we've already injected it into the eyes of our mice, and we know that this healthy version of ELP1 gets expressed in the retinal ganglion cells. And in fact, we're going to be injecting this virus, encoding this healthy ELP1 into our mice that have FD starting next week. So I also want to tell you that gene therapy has been approved for other genetic diseases by the FDA, and in fact has been shown to work really, really well for another retinal genetic disorder. Okay, the second project we're working on is to try to improve GI tract function and metabolism in FD. So I know you're all familiar with some of the GI problems that occur in FD, but there are also metabolic differences in FD. So for example, many people with FD are very thin, and in fact their BMI or body metabolic index has been shown by work in the Dysautonomia Center to be significantly reduced. And so the question is, why? And so it turns out metabolism, shown here, is very connected to what's happening in your nervous system and very connected to what's happening in your liver and to what's happening in your gut. And your gut is full of bacteria, trillions of bacteria. And these bacteria secrete chemicals that affect the nervous system. And so together, there's an axis called the gut-brain axis, which also involves the liver, and this regulates your body metabolism. And so we suspect that something is awry here. And so using, again, our mouse models, we're investigating this. And we're also investigating this using the stool samples and the serum samples that you have all so kindly been sending us. And this project is a collaboration. We're collaborating with Seth Walk and Valerie Copier, who are both colleagues of mine in Montana. Seth is a terrific gut microbiome ecologist, and Valerie Copier is a terrific biochemist with expertise studying metabolism. And of course, we're working with the Dysautonomia Center on this project and with all of you. And I'll show you some of the preliminary data on the next slide. Okay, so here's our analysis so far of the gut microbiome from the stool samples that you've sent us. So we've analyzed the diversity and the numbers of bacteria in the stool from people with FD and from their relatives. And there are two major findings. One, there are fewer types of bacteria. So that's referred to as diversity or alpha diversity in the microbiomes of the people with FD versus their healthy relatives. And there are also significant differences in the types of bacteria and numbers of bacteria in the relatives versus the people with FD. So something is very different about the gut microbiome in FD. We find the same thing in our mouse models that have FD. And interestingly, many of the patients are often taking antibiotics. So you might not be so surprised to find differences in the gut microbiome. But in fact, our mice don't take any antibiotics, and we're still finding the same kinds of differences in diversity. So this is something we're going to continue to work on. We'll be working closely with the Dysautonomia Center on this and try to figure out how this could be clinically useful to those of you with FD. Now, similarly, we find significant differences in the metabolism of people with FD versus their relatives. And we see these metabolic differences both in the stool samples you've sent us and in the serum samples. And you can see many metabolites are lower in FD and some are higher in both the stool and the serum. And I'll give you a synopsis of these findings so far on the next slide. So some of the major take-home findings we find is that in general, most metabolites are decreased in patient samples compared to relatives, except for choline and taurine, which are both elevated in the stool of FD patients, and urea, which is also elevated in the serum of FD patients. So why are these changes clinically significant? Well, choline is really important. It's converted to an important signaling molecule for neurons called acetylcholine. It's also converted into phosphatidylcholine, which is a key component of cell membranes. Taurine is important because it's used to make bile salts, which are critical for breaking down compounds in the gut and nutrient absorption. So the fact that it's elevated in stool and decreased in serum could suggest that there's faulty reabsorption of nutrients. Urea is a product of protein being broken down. And so the fact that it's elevated would suggest that there's too much protein being broken down in people with FD. And that would be expected if pathways for making energy for cells in FD were not working at the highest efficiency. In support of this finding, we also see decreased levels of amino acids in serum, which are the components of protein. And it's also supported by the fact that many patients have muscle breakdown. And again, that would happen if you're trying to make more energy. So in summary, what these data suggest is that proteins are being broken down in FD to obtain energy, and cells are essentially starving for metabolites, and that's why this excessive breakdown is occurring. And our data also suggests that there's some dysfunction in the absorption of nutrients in the gut. And so again, we will be discussing these data with the Dysautonomia Center. We're going to continue to work on this and try to see what we can do with these in terms of translating this to some kind of therapeutic. So that's it. I want to thank everyone, especially those of you in families with FD, because your samples have been critical for this work. I want to thank the Dysautonomia Foundation, the NIH, which has funded all of our work. I want to thank the fantastic people in Montana who have done all this work. Mike Brownstein, David Brenner, who first brought me into the FD Center, and my terrific collaborators in the Dysautonomia Center and working with me on the Retina Project. So thank you very much. Okay, I guess I can't comment on my own talk. So we'll move on to the next talk, where I'm really thrilled to introduce Dr. Miguel Weil from Tel Aviv University and Dr. Bat El Bar Aluma from Tel HaShomer.
Hi, my name is Miguel Weil. I've been part of the FD family since my eldest son nears birth 29 years ago. For the past 16 years, I've been in my laboratory at Tel Aviv University. We have been involved in understanding the role of ELP1 ICAP in neuronal development in animals and cellular FD models. Starting 10 years ago, we have been developing drug screening systems using patient cells by screening thousands of new or old drugs to find potential treatments for rare diseases. And as part of this effort, I am presenting you today, together with Dr. Batel Baraluma from Shiva Medical Center, a new clinical perspective for an old known factor in familial dysautonomia. And this old factor is called nerve growth factor, or NGF, which is a survival factor of the peripheral nervous system. This factor was discovered in the 60s by Rita Levi-Montalcini and Stanley Cohen, which awarded them in 1986, the Nobel Prize. And the fundament of this factor is that it is originated and produced by peripheral organs, which are nourished, innervated by this peripheral system, by these peripheral neurons that are completely dependent on this factor for their survival and function. Without NGF, these neurons will die. So what about NGF as an old suspect in FD pathology? So since the 80s, early 80s, has been shown that NGF deficiency drives to poor peripheral neuron development, function, and survival, as it is in familial dysautonomia. And also they found at that time that FD show NGF reduced activity generally, and surprisingly, they found that NGF levels are low in serum, blood serum from FD patients. This, at that time, gave us the hint that probably NGF-related problems are related with the genetic origin of the FD disease, which turned out not to be the case. But the problem today still, this paradox still remains. So, and the time we tried from the last, recently, we tried to find in our research to find out that there is a problem in NGF regulation of NGF synthesis and in FD patient cells, and to find out the connection to the serum pattern found in FD patients, we established a test to measure NGF in serum samples. So this brought us to the question whether is NGF a useful FD biomarker in serum, that which eventually will allow us to state the understanding whether NGF could be a useful factor for clinical assessment, to understanding whether connecting to data, patient data, and understanding whether this could be a marker to follow any future treatments. So to this end, what happens in FD, you could see in the left-hand side that the NGF levels are low in blood, and we think that potentially to test this question, we would like to find a drug that could have an effect increasing NGF, and concomitantly would be able to detect this increase in blood serum. So we managed to find in the literature such a compound, such a drug, which is a derivative of vitamin D. We tested this vitamin D on FD patient fibroblast cells, and it turned out that it works, that potentially this vitamin can increase NGF levels in FD cells, and that brought us to to testing vitamin, and the clinical idea that to test vitamin D3 and derivative called calcitriol potential to modulate NGF in serum of FD patients. But the floor is yours.
Okay. So many of you know what is vitamin D because you take it. Some of you take it regularly for to increase calcium against osteoporosis. So we have. We're going to talk about 3 forms of vitamin D. First, the one that you're taking. Some of you are taking. But to be active, it has to go through 2 stops in our body. The first one is in the liver, where it goes hydroxylation in a 25 spot, and then it is called 25-hydroxy vitamin D3. It is not yet the active form, just the semi-active. And then it goes to the kidney, where it goes through another hydroxylation, and this is the active form, which is called calcitriol, or 1,25-dihydroxy vitamin D3. So we know that we have a very good way to measure NGF levels in the blood, very reliable way, and we want to test if vitamin D can increase the levels of NGF. So we're gonna measure at baseline levels of NGF, levels of the active form of vitamin D, and also other forms related to calcium metabolism. Then we're going to give one of the 2, or the semi-active or the active form, for a period of 6 weeks. At the end of these 6 weeks, we're going to measure again levels of NGF and levels of 25-hydroxy vitamin D, and to see if indeed the increase in vitamin D caused the increase in the levels of NGF. Then a period of washout, and then again with the other compound. Hopefully we will get good results, and we'll see that there is a correlation between levels of vitamin D, active vitamin D, and NGF, and if the results will be encouraging, then we will move forward to an open trial, to see if there is also what are the clinical effects of increasing NGF in the blood. Thank you.
And to finish, I want to acknowledge the people involved in this research in my lab from the present and past, and as well to obviously to my sharing collaborators at the Shiva Medical Center, and as well our collaborators at the Dysautonomia Center in NYU School of Medicine, Horacio, Lucy, and Maria. Thank you very much.
Thanks so much, Miguel and Badel, for a very, very interesting talk. And you have a really powerful platform for discovering new compounds to help in FD. So that's fantastic. I want to thank all the speakers for sharing their very exciting research findings with all of us. I have a brief summary slide just to summarize what we've been discussing, and then I'm going to turn this over to Adrian Gilbert, who will moderate a question and answer session on current research. So you can see in this slide that we all share the goal, as Sue described initially, of increasing levels of ELP1 in cells. For any of this work to occur, sorry, my phone's ringing, we need a natural history study and a clinical development plan. And that's what Lucy and the Dysautonomia Center are working on. The study that Miguel and Batel discussed about increasing vitamin D levels to increase downstream signaling molecules like NGF is a very powerful strategy. The work Sue described with her great team, including Elisabetta and Monica, designing small molecules that increase splicing so we can increase levels of ELP1 protein is going to be extremely powerful. The work we're trying to do with gene therapy, we're hoping will be very effective. It's a collaboration with Sue's group and with Claudio Punzo, who's a retina gene therapy expert at UMass. And lastly, we're hoping to sort of look at the whole big picture too by looking at metabolism and the gut microbiome and see if things can be tweaked there to overall improve health. So I just want to thank everybody. It's a collaboration. We all need to work together. And I apologize to all the researchers who weren't included in this, but believe me, there's a lot of great work going on in research in FD. Adrian.
Great, thank you, Francis. This is fascinating and I'm really enjoying it. I think now what we'd like to do is to put some questions to our distinguished panel, who I hope will be able to answer at least some of them. And together with some prepared questions we have, we'd also be very happy to have new questions if you could post them either in the YouTube live chat or to the FD website. We'll be very happy to try and include them. And I have about half an hour or so to be able to deal with your questions, so please don't be shy. So let's start. I think I'm going to start first with Lucy. I'm going to be with some questions which have already been posted. Lucy, how's the natural history study going? And is it useful to you? So this is your chance to tell us what we need to do.
Yeah, thanks, Adrian. I think that the natural history study is going well in terms of what we've been able to set up, what we've been able to put together, and what we've been able to build. We just need a mechanism to make sure we're capturing more people. I mean, the data becomes more powerful and more useful the more people we have included. I mean, it's this incredible resource for us that has shaped clinical practice and also research. I mean, we have been following, for example, things like the retina, like Frances mentioned in her talk. We discovered a number of years ago that the reason people with FD were losing their sight was because the nerves at the back of the eye were dying. So what we actually are able to do with the natural history study is track those nerves by measuring them over time. So really that gives us a target to be able to see if drug therapies that we're trying are working. And if they're not, that means we need to go back to the drawing board and we need to think again in other strategies. But the natural history study is really the backbone, I hope you see, to many of these basic science projects. It's like I mentioned to you, like basic science can't happen. It can happen in a vacuum, but it's less effective if it happens in a vacuum without the consideration of the clinical data alongside that. And it's when you really bring clinical science and basic science together that you get this translational element that helps you actually progress and develop new therapies. So
excellent. So it's going to help. It's going to help us for all of the approaches. Yes.
Absolutely.
Sorry to interrupt you. So that's great. So it's really, on one hand, we're going to learn everything that we can about what's going on, but basically the future, when we design the clinical development of whatever is going to come out, some of these ideas that we've come out, this is going to serve us to move forward. So it's really, really important. It's just going to take much longer if we don't have a good natural history database. So now, Francis, we have a question for you. So how close are we to treatment for retinal neuropathy? You've whetted our appetites, and someone's even asking, how about a gene therapy cure for FD? So how long have we got to wait?
So again, there's several ways to approach the problem of trying to rescue the retinal ganglion cells. Sue's working on her splicing compounds could very well do the trick, and that's part of the project her group, and Elisabetta Marini and me and Claudio Punzo are working on together to see if those compounds will work, in addition to replacing the gene and doing a classic gene therapy. So for the mice, we were held back a few months because of COVID. We'll be doing the injections next week. We'll know in a few months if it's working in the mice. If it's working in the mice to protect the retina, then we will start having serious discussions as a community with the foundation, with the Dysautonomia Center, as to how to move forward and translate this to patients. That's one thing. In terms of beyond the retina, if it works in the retina, then what would be fantastic is to deliver the gene systemically. So, for example, there's another genetic neurological disorder called SMA, and gene therapy has been shown to work systemically there beautifully. And you just do one injection of the virus, and infants are able to, who were practically paralyzed, are able to hold themselves up and to move. So we would like to consider doing systemic gene therapy, but the proof, first test of principle will be our experiments in the retina.
Yeah. I mean, this is amazing. When we read about gene therapy in all of these other diseases, and we always get a bit jealous because we think, why can't someone do something in FD? And it looks like maybe we are going to do something in FD. I mean, we can't promise, but we're doing it really, really trying very hard. We've got the best people on it. And the idea of being able to save people's sight, people who are watching this, we're going to keep being able to watching this going forward. I can't think of a better thing to be doing. So thank you for that.
I'll mention one point just to also boost people's spirits. The NIH has been funding all of this work. It's been funding Sue, it's been funding our lab. So, we might feel like we're an isolated community, but if we make good, convincing arguments to the NIH, they support us.
Yep. And we haven't really said it, but the people we've collected here, and yourself included, are really quite good at working with NIH to get the funding. And that's as important as the science, because if they don't fund it, we can't do the science. So this is a really, really important aspect as well, and I'm glad you mentioned it. If anyone, by the way, has questions about anything that we're saying, please, again, this is my plug, write them in the live chat or send them in by one of the other media, but we'd be very happy to try and answer your questions. I have one or two, but for now I'm going to keep going. So the next question is actually about new therapies coming down the pipeline, maybe some of the things which haven't been mentioned up to now. And I guess we should divide these between symptomatic and really disease modifying, like gene therapy. So maybe if I could ask you, Alberto, we could talk a little bit about the symptomatic treatments. In particularly, maybe people are interested to know about blood pressure and how we can stabilize blood pressure in FD. Maybe you can give us some thoughts about that.
Sure, absolutely. I'll be glad to. So one approach we've been using for a number of years now at the center has been the carbidopa, right? Which is familiar to many of you, and Dr. Kaufman mentioned briefly in his initial presentation about clinical updates. Well, many of you participated in a clinical trial for which we used, in which we used carbidopa to improve blood pressure variability in patients with FD. Well, that trial finished a few months ago. We put together all the results, and we are excited to announce that the carbidopa works very well. I mean, it's now absolutely proven, right? And we did a very rigorous trial using even placebo, right? Many of you don't like placebo, but we trial led by Lucy, and as I'm telling you, it does work beautifully compared to placebo, and the results will be published very soon in a great journal called Hypertension. So this is going to be publicly available to everyone, and it's a great example of how placebo-controlled studies, placebo-controlled trials in FD are feasible and how we can define whether a drug works or not with these mechanisms, right? So I want to specifically congratulate Lucy about this, but also congratulate all our FD patients who participated in that trial, because now we have solid results showing that carbidopa works for them.
That's very important. I think one of the things I know for myself, and I may be speaking more as a parent here, we're always a little bit nervous to start new treatments because we're going to discover things that these treatments are going to do with our patients or our kids which we weren't expecting. And I think it's really important to hear about, in a controlled way, the safety of these new treatments in FD. So carbidopa, it's safe?
It is very, very safe. You know, it's we didn't find any severe side effect. Some people experience some mild, very mild, short-lasting discomfort, particularly a gastrointestinal discomfort, but overall, it's one of the safest medications that we work with, right? So nothing to do with other medications. It doesn't have any impact on respiration, and overall, it's extremely safe.
Excellent. So it's very well tolerated. That's good news. Okay, and it's been proven, and I look forward to reading that. Okay, so now when we're talking about other therapies, we're going to talk a little bit more about those disease-modifying therapies. And Sue, I just add on a question which came in. So actually, how does increasing ELP1, how does that help neurons? And what can we expect from increasing ELP1 to help FD patients? This is an excellent question, because the disease isn't ELP1, it's FD. So exactly. How could you answer that?
I actually saw that question come in, and I'm going to punt the what does increasing ELP1 do to neurons to Frances, because she could tell you that much better than I could in terms of like studying neurons and looking at that. So Frances, do you want to take the first part of that, and maybe I'll take the second?
Sure. Okay. So ELP1 is really an important protein in the cell. So, you know, your cell, you have all these little departments within the cell, and all those departments have to communicate well with each other in order for that cell to be healthy. You know, it's like you need your liver to work and your kidney to work and your heart to work. So the cell has all these essentially internal organs as well. So what ELP1 does is it is involved in the production of other proteins. So imagine like a waterfall. If there's some interruption of the waterfall at the very top, it's going to have a huge downstream consequence. And that's what's happening with ELP1. If there isn't enough ELP1 at the top of the waterfall, then many other proteins aren't going to be produced normally. And if you're missing all these proteins, then many aspects of the cell biology aren't going to function normally. Also, what can happen if you don't have ELP1, you get big clumps forming inside cells, aggregates, and that can also gum up the cell so it physically can't function normally. Right, Sue?
Thanks, Frances. Thank you for that great explanation. And I will just add to that about what we know about what happens in our mice when we increase ELP1. So as I mentioned, when we've increased ELP1 by feeding mice kinetin and by also using other compounds, and when we show that there is increased ELP1, the mice walk better. So they have improved gait, they have improved balance. We do things, we test mice on we make them walk on a gait analyzer. We make them walk put them on a rotorod, a little rod that spins, and see how well they can stay on the rod. And we can actually count the number of neurons in different areas of their peripheral nervous system. And all of these things are improved when we can increase ELP1. So of course, we don't know yet what this will look like in FD patients. But as soon as we get a drug or another method like gene therapy or something that's ready for patients, then we'll be able to find out.
That's exciting. That's really exciting. So I suppose when we get to the clinic, that's exactly one of the things we want to validate, is to track the increase in ELP1 in the patient and to track these changes in their disease or in how they function going forward. So this is really what the stage that we'd like to get to when we get to the right treatment to go into the clinic, given that it's safe. Okay, thank you, ladies. That was a great answer. Go ahead.
I would just like to add that we can now track the levels of ALP1 in FD patients in their blood, and this test was developed with help from PTC, and so now we'll have that test available to us so that when any of these treatments come on board, one of the things we will be able to test is the levels of ALP1 in patients' blood.
Okay, excellent. So ALP1 is something to be followed up. I'm trying to scribble down all the time new questions which are coming in. So now I'm going to put a question actually to Miguel out here in Israel with me. Miguel, we actually heard from Francis that she's taken patient samples and she's done some great things with Biosome and looking at retina as well. My question to you is, you want samples too. So if we get the samples in Israel from patients and through the treatment center, what's the research that you're going to do? And I understand, maybe tell us what you want to do with those samples.
So, okay, so first of all, I need to tell you that some of the samples we use for the testing of this NGF in serum were given by the FD Center in New York, and that helped us to understand the levels in blood. Now, why do I need cells? We need basically very small amounts of skin biopsies, about 2 millimeters in size, from which we will take a high number of cells, where we are going to use in our pipeline to test future drugs, as well as to understand in a personalized level for each patient what is the phenotype, how the FD comes out in cells like skin fibroblasts as well as in neurons. Once we have that, we would like to test the effects of the coming drugs in the pipeline, whether ours or those coming from Zoo, and test on a personalized basis the effects on that cells, and hopefully we could go back with the information to you and into the natural history of each of these patients, and probably predict the way these drugs could be given specifically for those patients that we were work on the cells previously. Okay. But this is kind of big picture.
Okay, so that's a great answer. So really what we're talking about is, although we can actually see in front of us personalized medicine, but we can see in the front of you, I take myself out, there's the 5 of you and other people, but we're really building a network of how to exchange the information and build our understanding of which are the molecules that we want, the drugs that we want to try going forward. We'll test in animals, but we're going to share the information and hopefully get into the clinic soon for all of the patients. So that's a really clear message that's coming out. I'm going to move on to another question now, and I'm going to throw it actually at Sue. And it's a very basic question, but I think needs a quite complicated one to answer. Why do different people with FD have different clinical profiles?
It's a very interesting question and an important question. And I want people to understand that this is typical of genetic diseases. So most people with genetic diseases, the phenotype is not exactly the same from patient to patient. And so in FD, if we think about it, while everyone has the same mutation, that same change that you've heard many of us talk about, the rest of people's genomes is not identical, right? They have many other different genes that are different from each other. And so many of these genes likely modify the phenotype of FD. And what that means is some of these genes may have certain what we would call certain alleles of genes that maybe make FD more severe, maybe make FD milder. I'm sure that some of the phenotype of FD between patients also differs by the extent and quality of medical care that we received earlier. But we're focused on understanding what these genetic differences are. So one of the goals of future work would really be to be able to understand what other genes modify the phenotype of FD. So what are the other genes in the genome that make some patients have a very severe phenotype and others be relatively mild, even though their FD mutation is identical? And we don't know the answer to that.
That's a really good question. So what are the other genes? And actually someone has just written in and asked us, do you think that the level of ELP1 in a patient, is that indicative of the severity of their disease?
So our hunch, I think that everybody here would agree with me that that is our hunch. We believe that that's likely true. We think that it makes sense to us that if you don't have as much ELP1 as others, you can see from the diagrams that we showed that when there's different levels of that exon 20 inclusion and different cells include different amounts of it. And so therefore it makes sense to us that some patients who are milder might be able to include exon 20 a little more often than others. Maybe there are some of these genetic modifiers that I talked about. Maybe there are other genes that are involved in splicing that are having an effect. We don't know at this point, but that makes sense to us. We have tried to look, and this is part of the goal too of what Lucy talked about in terms of the natural history and really understanding phenotypes in FD patients, because we would like to see if we can figure out a way to correlate the level of ELP1, now that we can measure it in patients, with specific phenotypes to see if there's a correlation between the levels and specific phenotypes. And we're working on that now with the FD Center.
So when we see a treatment increase the level of ELP1 in a patient, we want to know, is that going to help their disease, or is that just something that we can see as a measurement which isn't related? That's really important. We think it, but we need to understand it and we need to show it.
Exactly. Exactly.
Okay, great. That's an excellent question. I've actually been asked a question which I don't know I'm a good person to answer, but someone's taking a lot of carbidopa, up to I think more than 20 capsules a day, and it's working great. And how do we work with the pharmaceutical companies or the compounding or pharmacies to be able to see how maybe we can get higher doses made and to help our patients? And I think, I mean, I don't know, this is necessarily a research, more a pharmaceutical question, but it's probably something maybe the compounding pharmacists in the hospitals could perhaps help with. Do you want to mention, Alberto, maybe do you want to comment to that?
Sure. I think the problem lies on the fact that the commercially available capsules for carbidopa are 25 milligrams, right? So that's what most pharmacies have, 25 milligram capsules. The dosages we are using in FD patients to treat the blood pressure problems, the dosages of carbidopa are much more high, much higher than that, right? We are using in the range of 100, 200, or even 300 milligrams. So that results in the fact that many patients are taking a lot of capsules. So, Adrian, as you mentioned, the best step or the best course of action is probably to get in touch with a compounding pharmacy who is able to customize the dose of that patient, and instead of that patient taking 2 or 3 or 4 or 5 capsules, maybe they just have to take 1 or 2. And from the FD Center, we are glad to help because we have this experience with other patients who are having this predicament, and we can help, particularly if they are local. In the New York area, we have a number of compounding pharmacies that work with us.
But also there are compounding pharmacies in the other hospitals, even when they don't come to NYU or Tel Shomer, and you can maybe help and be in contact with some of those centers as well, and to reach out. Yeah. We've actually had a question which relates a little bit to the gut research from Francis. The people are asking, should we be taking nutritional supplements that include taurine or choline? Is this something that you think would be helpful, or should we wait to see if there's more research?
First of all, I would wait because we're still analyzing all the data. We're still analyzing all the data, so I would wait. But certainly the goal is that we be able to design some kind of supplement, recommendation for some kind of supplement that could be beneficial based on this type of analysis. I guess I should also mention that, part of the reason why there is this protein breakdown, we think, is because energy production in the cell isn't optimal in FD, so that there's a part of the cell that makes energy, the mitochondria, and what we know now is the mitochondria don't work very well. So we're actually doing some experiments with supplements that bypass the part in the mitochondria that doesn't work. And that's looking promising in mice. But again, it's early days, but definitely that's the end goal, is once we've completed all these policies to have some recommendations.
And I guess it will be the same, Miguel, when we're thinking about calcitriol or even vitamin D3, that we're sharing the research, but it's a little early to go and to tell people to go off. And I mean, we're taking vitamin D, but whether specifically what you mentioned, calcitriol, and what dose, it's a little early, but this is something that we'll try to come up with as quickly as possible with a recommendation. Would you agree with that?
Yeah, sure. I think that the most important point that I want to make is that we know that a lot of people, a lot of children and FD patients are taking already vitamin D supplements, but the idea here is to clarify and specify how certain amounts of vitamin D derivatives could increase these levels of NGF, and probably because it has been shown that this has a problem is related with FD, probably bringing it up, it will have an impact in the nervous system and hopefully will increase survival of those neurons that are compromised by the lack or the low levels of this factor. And in this compensatory way, we might probably have a beneficial effect of anyone that can be taking these safe vitamins in a very well distinguished manner, right? In a very directed manner.
So someone's asking us: are ELP1 and NGF related?
This is an excellent question.
Okay,
can I answer?
We can agree. Sure.
So no, they are not related per se, but they are connected in a way, like Francis mentioned, that there is kind of this waterfall coming down, related with the ELP1 mutation. I mean, the FD mutation has an impact on many different systems, including the production of NGF. We still don't know what is the relationship, but we know that if we could build this up, probably some things that are related with NGF can become better.
Excellent. Well, I think we've actually answered all the questions which were put to us, both during the week and today. So thank you, everyone. So some of them you were prepared for, and some of them you were not prepared for, and I think you've done extremely well in managing them. I want to I think it's been exciting, uplifting, insightful. No one should get carried away that tomorrow we have that treatment, but we're working very, very hard with some very good people to bring that forward. And I think it's just really great to see such a diverse and global panel together, acting and working together. So thank you, everyone, for your presentations, for your answers, and for your time, and for everything that you do for FD. And now I'm going to hand it back basically to Lainey to wrap up today's event.